So we're going to open up to questions. We have a few questions here on the table and some coming through on the iPad. First question is to you, Nicole, which is from Matt Evans. Do you have any experience of how nanopore sequencing handles DNA from formalin-fixed tissues?
Could you repeat the question?
So could we use nanopore sequencing to look at DNA from histological specimens from fixed tissue?
Do I press here?
No, just speak.
Okay. Yes, I believe you can. The problem is that, not the problem, like the step you have to do beforehand is to extract the DNA from the tissue, remove it from the tissue so that it is then in solution, and then it can be sequenced and used in nanopores. So you can use it as long as you remove it from the tissue, basically. Okay,
great. Thank you. The next question is really to the panel, and it's from Dr. Feliciano. And the question is, and I'll add my bit to it. The question is to the panel. Some published observational data suggest that gender may be a prognostic factor for cholangiocarcinoma, with females potentially having a slightly better prognosis. Could you share your perspective on this? And do you think there could be a biological basis for this finding? And I'm going to ask Veronica to start off first, because the cell line that you use, CCLP1,
yes,
is a human cell line,
yes,
from a 48-year-old female, Caucasian, who had intrahepatic cholangiocarcinoma. So what are your thoughts on that initially, about the gender issue and maybe females having a better prognosis and biological basis?
Actually, yeah, could be probably the difference about the gender. I mean, in terms of protein expression that you can use for biomarkers. And actually, in our study at the moment, we could make maybe later just to understand if the overexpression of our 3 proteins in a tumor microenvironment could be useful also to determine to classify the patients based off their gender. Maybe could be an idea for that.
Okay. Stephanie, any evidence of the preneoplastic lesions behaving differently between genders?
That's a very good question, but actually we have not really asked this question before, so I don't know. I cannot answer it, at least for the preneoplastic lesions. We do know from hepatocellular carcinoma that there is definitely an influence of gender, and there's also more research going on. So maybe this is something lacking in cholangiocarcinoma, and I'm not aware of any studies really looking into that in detail.
Okay, I'll move on to Mark. Mark, does your BRCA splicing prevalence vary by gender, and also I'd just throw in there by any other demographic factor, and also did it vary by the prognosis of those patients and how well they did post-resection?
So there was no difference between BRCA mutation rates on the basis of gender. From our data sets in Liverpool, certainly in the resected setting, there is a propensity for males to do worse from our data sets. From the biological data that we've generated, there is what we've seen in the single cell setting certainly is that estrogen receptors and genes related to estrogen receptors in the various estrogen pathways are upregulated. So it wouldn't be surprising that there is a role, that gender may play a role in impacting outcome. But as our colleagues here have said, I'm not familiar with any published data to suggest that definitively.
And in terms of prognosis, how those patients did post-resection, survival, recurrence, would that correlate with the BRCA splicing?
I think it's based on the numbers that we have. It's very, very difficult to extrapolate much in terms of the survival analyses based on the numbers that we have, and also given the actual frequency of the mutations within the cohort. So when we ran those initial survival analyses, we didn't see that we could generate meaningful statistical differences at this point in time. So there is a need that if we start to see that this signal is preserved functionally, at the functional level, I think we'll need to think about validating that on a larger, validating these mutations on a larger, much larger series, across the UK or beyond.
Okay, thank you. Simon?
I suppose, I was just going to ask, for the BRCA reversion story, can you definitively prove that? Because equally it could just be a deleterious mutation, that cell population's dying out, and that's why it's going down, and it's very, it's a rare allele frequency and a low cell population number.
Yes. So in terms of our functional plan going forward, we'll be using a host of different cell lines, isogenic cell lines and things, patient-derived as well, in our splice model. And what we would aim to do is feed in each of those mutations at differing time points in isolation and combination, and then treat those over time with the various therapeutics involved. But a lot of this is going to focus around small interfering RNA and various approaches around that, so Cas9 things. So that's going to be our plan. The difficulty with this work is that the spliceosome is such a heterogeneous phenomenon in and of itself, with multiple different genetic and epigenetic factors in play. Ultimately, we don't believe this is going to be a process which is affecting all cells within that population, but it may be affecting enough cells to generate a degree of instability which is potentially a window of opportunity.
Can I ask one additional question? So just coming back to the nanopore, do you think your choice of sequencing platform has limited your ability to do what you want to do ultimately? Can you do multiplex with other sequencing platforms that don't have that problem when you put human serum on there?
Potentially, yes. We are ultimately a nanopore group, so the choice was also given to that, and with the collaboration of the group with Oxford Nanopore. One of the advantages of the nanopore sequencing devices is that they're portable and they do not require big or slightly more cumbersome and more expensive equipment, for example, the Illumina sequencing or other types of next-generation sequencing. So in a way, yes, it has limited it, but at the same time, it brought advantages that we wouldn't have had with other types of sequencing. So it was...
There is a danger you can lose your proof of principle that could work as a classifier by limiting yourself to a particular sequencing technology, but...
Potentially, I guess when you're at the forefront of trying to develop new tools that sort of break the ground, you choose a trade-off. So we worked with that because we believe that it could help, for example, once we sort of refine the detection method, it could help with, biomarker validation and aid other types of research towards cancer. And also the versatility of the platform. The barcodes I designed were aimed at microRNAs for HCC or CCA, but by changing the targeting region, you can target any other microRNA that you want. So we chose those advantages, let's say.
Can I just ask a follow-up question on that?
Yeah.
Can you quantitate the level of your element, whatever you want to look at? Because,
yes.
You said something very interesting that is portable, so potentially it's clinically applicable. But if you think about in the future, you want to assess prospectively, maybe you have one microRNA, two microRNAs, and then you don't have the comparison with the rest of the group. So can you do that with your nanopore technology?
Yes, so I showed that with the calibration curves that we built, we are able to sort of reverse engineer and go, okay, these are our calibration curve. We got this type of response, so we expect that the corresponding amount of target is within this this range. which, I mean, in the future, when things work perfectly, this could help also quantify the level of microRNAs present in the samples that we test and potentially differentiate. I don't know, we see that this type of microRNA is within these levels when the patient has gone resection and things like that.
Thank you.
Well done, Nicole. I was your supervisor, so I'm trying not to say anything, but you're doing really well. John?
Back to BRCA. Mark, I didn't get a feel for whether you thought these splice variants were in fact...
Yes, we believe they are pathogenic. So when we went through each of them, they're all pathogenic. So those were all of the ones that we then also took forward to do PCR on individually. And the most commonly occurring pathogenic variants did appear to impact the gene expression in the tumour compared to the normal bile duct. So that was replicated across a number of the samples, yeah.
Okay, thank you. And consequently, what therefore would be the selection pressure to desplice, to revert? What's going on?
So this is where we feel that it is an early event, that as time progresses, it would ultimately become detrimental to have no BRCA functionality at all. And is that hence why we're also seeing that the protein is in fact present across quite a few of the patients still. So that selection pressure, we feel that it's an early event that is then evolving over time, and that it's a dynamic event. And these reversion processes have been seen in the breast setting with some of these specific mutations. But at this point in time, it's extremely, there are so many factors going on within the spliceosome itself, so many various epigenetic and genetic factors. It's very, very difficult to tease out the individual impact of each one of these. But we think that this is just the first part of a potential story here that is worth exploring.
Yes.
Chiara.
Stephanie, I have a question for you. You looked at the preneoplastic lesion through different technologies with DNA sequencing, methylation, and gene expression, and you made the case that some of these lesions might progress to cancer. I was wondering, do you have any data, or do you think there is a scope to get any data about genomic signatures, more kind of a nucleotide signatures, to understand if there is any carcinogenetic exposure that, causes these preneoplastic lesions, and then potentially we can use this preventive strategy for cholangio?
That's a very good question. So what we are using is FFPE tissue. And with this tissue, it's very difficult to working with genetic signatures because the fixation is causing mutations or it's looking like mutations if you sequence the FFPE DNA. And so we haven't really looked into that. But we are trying always to look at the etiology also for the patients. But unfortunately, this is not possible in all patients. So like also shown yesterday, for more than half of the patients, we don't know the etiology. And this is making it hard. Another limitation is that most of the patients, they had surgery because of the cholangiocarcinoma. So there is already the invasive carcinoma, but we don't have a group of patients that have only the preneoplastic lesion. We have only very few patients that have only the preneoplastic lesion, and that was because there was suspicion of cholangiocarcinoma, but then it turned out it was caught so early that actually there was no invasive carcinoma. So I think there's a need of increasing the number of samples with only preneoplastic lesion to really understand that better.
Thank you.
Okay, so last question, and 1 minute to go. Simon,
Just coming back to the BRCA story, I mean, obviously now there's a lot of work on mutational signatures, and it would be interesting to see if you see a BRCA mutational signature in those ones that you find where you find the deleterious mutations.
Yeah.
I don't know if you've had a chance to do that or thinking about that, because I'm not aware of any publication on the mutational signature. Just there's a BRCA signal in perihyla, but it might be interesting to see.
Yeah. So the difficulty with the signatures is that typically a lot of them, again, are referring to the more commonly reported types of mutation. So I'm not familiar myself with how much splicing is incorporated into those signature generators, but certainly something that we'll explore. Thank you.
Okay, so thank you. Thanks again to the panel. Amazing, really interesting session.